Depending on the pathway analysis approach that you use to analyze your data, that can have a dramatic impact on the type of insights that you're able to derive from your results. Let's look at how these different analysis approaches differ from one another. The first and most common approach is what's called overrepresentation analysis. It's most commonly referred to as enrichment analysis. This is a very simple but powerful technique that leverages simple statistics to identify pathways that may be significant given your data. The other benefit to this particular approach is that it doesn't require very much input data. You can submit a short list of genes that you're interested in, and you'll get a list of pathways that are enriched for that set of genes. The downside to this method is that it makes an assumption on the background of those genes. Because you're only submitting a short list of genes, it has to make an assumption of what is the denominator. For example, if you have 100 significant genes, the system might not know what was the total number of genes that has been analyzed. In other words, is it 100 significant genes out of 10,000, 20,000, or is it 50,000? So it makes some assumptions on that. The other downside to this method is that you end up throwing away about 90% of your data. For example, if you've done an expression experiment where you have observations for 20,000 genes, usually there's only about 1,000 or 2,000 that you're interested in, out of that entire set. That is a major disadvantage because you spend a lot of time and money in getting those measurements. The biggest limitation of this method is that it assumes that all of the genes in the pathway are independent. It completely ignores the interactions among genes within the pathway. This can lead to a lot of false positives. A slight improvement on enrichment analysis approach is what's called functional class scoring. You may have encountered gene set enrichment analysis, or GSEA, which was pioneered by the Broad Institute. In this particular model, it is more accurate than the simple enrichment model because in this method we're going to use the entire list of genes that were measured. In the case of gene set enrichment analysis, we will also supply the p-values and supply them a ranked order. Depending on the type of functional class scoring method, you may provide some weighted scoring based on those p-value. But because this is still based on an enrichment model, it is still going to ignore the interactions among the genes on the pathway. It is still a count-based model, and as a result this method can lead to a lot of false positives. In the mid-2000s, a completely different type of approach was proposed. Its goal was to mimic how we think about pathways from a biological perspective. In this case, we're trying to identify how perturbed a pathway is given a particular input. We can do that by leveraging the topology of the pathway. In this particular method, we're going to consider each gene's role, their position, the magnitude of change between our two phenotypes, and the interactions among other genes in that particular system. One of the advantages about this particular model is that it's able to predict what's going to occur in that system given the observations from the data. However, there are also some downsides to this method. First, it requires more data in this particular model. We need to have measurements for all of the genes between the two phenotypes, their measured fold change, and their p-value. When this method was proposed, that was a very difficult and expensive task. But with advents and improvements in transcriptomics and proteomics technology, and bioinformatics, it is a much easier to accomplish. This method does take slightly longer than the enrichment models. The enrichment models can be calculated in the matter of seconds or less, but the topology-based model is very computationally intensive and takes anywhere from 5 to 10 minutes to calculate.